Next up, we are joined by Dr. Richard Mollard, the Chief Scientific Officer of clinical stage drug developer Pharm Aust, with the ticker code PAA. To tell you more, I will introduce you to Richard.
Thank you very much, Jane.
Let me bring up your presentation for you.
Thank you, Jane. So Pharm Aust is repurposing monopantal as a cancer, neurodegenerative and antiviral therapeutic. So repurposing means that we're lowering the risks and costs of development. Next slide, please. So this is a company snapshot, and all of these slides are available under PAA at the ASX. So I encourage you to interrogate them a bit further there. So how is this all possible that monopantal can start as a sheep drench and have applications in cancer, neurodegeneration and viral infections? So this comes under the umbrella of repurposing, which is common in the pharmaceutical industry. So here we have a sheep drench, which you see on the bottom left corner, hits the acetylcholine receptor, kills the worm outlined in black and also the larvae. It's specific for these worms. These receptors don't exist in sheep, and they also don't exist in humans. Pharm Aust was able to demonstrate off-target activity against a molecule called mTOR, or the pathway of mTOR signaling. So mTOR has interesting origins in Easter Island, which is kind of fun to look up. But inhibiting mTOR, which monopantal does, the signaling pathway, this induces autophagy, which has applications in cancer, where the cancer is pretty much instructed to destroy itself. In motor neuron disease, you can see here on the right, motor neuron, and inside the cell, you have misfolded proteins that accumulate and affect the function of the cell. So inducing autophagy helps clear those misfolded proteins. And for COVID infections, for example, as an antiviral or HTLV1, these viruses hijack the autophagy machinery to form capsules and hide inside the cell, and then be released to cause secondary infections. And monopantal competes with the virus to say, look, you're not going to use this machinery and we will destroy you and eliminate you from the cell. So on the left are some famous examples, Viagra, tamoxifen and exenatide, where repurposing has generated extra value in other indications. So Pharm Aust in this endeavour is protected by 5 patents, the 5th one to be awarded still. And they have a long runway, and you can see at the bottom coronavirus and HTLV1, if granted, expiring in 2040. So to get into the clinic, Pharm Aust had to take a drench, which is given to sheep, and which is associated with an exceptionally high safety profile, which is available under a public release summary because it's registered as an anthelmintic by the APVMA in Australia. However, the problem is the drench doesn't taste so good, and the liquid the drug active monopantal is dissolved in has a very poor taste. So we were able to take the active, and in the bottom middle you can see our convenient at-home tablets that are pressed. For first proof of concept as an anti-cancer drug, this is the latest data from our phase 2 trial in pet dogs with B-cell lymphoma. On the left, you can see the participants, their numbers, and next to that, they're arranged in ascending order of plasma drug levels attained. You can see we tried quite a few different doses to get the optimal dose. At the highest dose, we see some or minor weight loss in the dogs, which wasn't a concern to many vets, but to some. So we call that an adverse event. But if you drop the level of blood in the blood a little bit, we see no adverse effects of weight loss with plus or minus 2% of weight or changes over the 28 days. So a very safe drug. Next to that, we have all these green and red squares. So outcome observer is what the vet and the owner sees. Outcome veterinary is what happens if you interrogate with X-rays or ultrasound. So at the very bottom, you can see a lot of red, and there's a progressive disease in these dogs who do not actually get enough monopantal. Then above that, you see there's a lot of green. Where there's red, it's quite ambiguous or marginal. And this is because vets will disagree because the changes are so minor, where some will say, well, look, we don't see any change. And some say, well, we do. Some say this change may not be attributable to the drug. It may be attributable to the disease. And other people say the opposite. So we have ordered these in ascending order of plasma levels. And in the middle, you see quite a good spot where there are really no adverse effects. And all these dogs really have stable disease, at least 5 of 7 by interrogating with thoracic and X-ray or imaging. So we're taking this dose forward into a phase 3 trial, which we're planning now. We were fortunate enough that several of the veterinarians thought the monopantal was working so well that they would keep monopantal going and give prednisolone at the same time. And we were able to follow up to 6 dogs off trial. They all did quite well. 4 of them did very well with over 100 days or greater than 14 weeks, and some still going on. And this is compared to standard of care prednisolone alone, which usually gives 4 to 6 weeks. So the dogs on trial had high quality of life. You can see the dogs on the right going camping with their family, playing in the park with their dogs, and they had very good longevity. Although with prednisolone, this is retrospective analysis and we will still analyse it, but we're very encouraged by this data to go forward into a phase 3. This is compared to alternative standard of care as well, CHOP chemotherapy, which might give 26 weeks, but is associated with a lot of side effects and has a high cost, around $10,000. So the dog's hair might fall out, they vomit, they may defecate on the carpet. The kids cannot play with the dogs because the chemotherapy is toxic. So we think combining with prednisolone, for example, is going to provide a tremendous value proposition. And we're also interested in combining with CHOP to see if we can improve the quality of life. As far as motor neuron disease goes, on the right-hand side, you can see the brain with the nerve cell, and this innervates the muscle. And when these proteins build up, unfortunately, these individuals lose motor control. So they lose locomotive ability, speech, swallowing, eventually respiration. And unfortunately, life expectancy is very short. It is a rare disease, 1 in 400 people, and there are no cures. But you see the middle bottom picture with the green semicircular structure, which looks a bit like a Pac-Man, and it's eating the purple and blue misfolded proteins and ingesting them and then eliminating them out. And this is how we hypothesize monopantal is working to stimulate elimination of these misfolded proteins and offer protection to individuals living with motor neuron disease. We're very fortunate to have been awarded $900,000 by Fight MND for our phase 1 trial. You can see Neil Danaher in the top picture there, who is the co-founder and proponent for Fight MND. And we'll be undertaking this trial next year with Susan Mathers and Dominic Rowe. Next slide, please. So also we're looking at antiviral applications. So once again, autophagy. And on the top right-hand picture, you can see the life cycle of a virus, where on the left, the virus enters the cell, it releases its RNA, it replicates, subsequently gets packaged and then is eliminated from the cell. Our preclinical data on the left, where we show monopantal protects against cell death from SARS-CoV-2 in vitro. It reduces viral RNA load in the culture media. And in 2 laboratories under similar conditions, we see a reduction in viral secondary infection. In the 3rd laboratory, which had very different conditions, we didn't see this reduction, but this enabled us to come up with a hypothesis that monopantal is acting later in the life cycle of the virus. You can see here where the packaging goes on prior to release. So we're testing this hypothesis looking in HTLV1 infection as well. So we're lucky enough to work with a group who has fantastic preclinical models for looking at the mechanism of action in the life viral cycle. So you can see the bottom right here, those green semicircular objects again. This is what the virus hijacks. Monopantal will say, no, you're not allowed to take our machinery and we're going to eliminate you through the normal elimination system. So we're looking to set up these clinical trials as well next year. So this is our final indication for human cancer. As people might be aware, we conducted a successful phase 1 trial at Royal Adelaide Hospital, where we demonstrated stable disease in 3 or 4 patients tested. Unfortunately, the bad taste of the original formula meant we stopped the trial early and hence made the tablets, which we will now take into phase 2. We were able to show that we could inhibit mTOR signaling to induce autophagy. Adverse events were mild and the immune system wasn't compromised, which normally happens with chemo. So with monopantal, the immune system presumably can still fight the cancer itself at the same time. We are also able to get very good blood plasma drug data to take into phase 2. We have several physicians who are very interested in our combinatorial work, you can see in the graphs in the bottom right, with gemcitabine and cisplatin. So gemcitabine for pancreatic cancer, cisplatin for gastric and esophageal cancer. In each graph at the top, that's control with no treatment. In the middle, those 2 lines are single treatment. The bottom line is combinatorial treatment, and you can see how effective it is. So we're looking to set these up next year. So these are the timelines we're targeting. And as I said, these are available on the internet at the ASX, and I'm probably running out of time. So I would encourage you to look at these there. And final slide, please, as well. This is the management team who is responsible for this project. So thank you for your time.
Richard, thank you for the great presentation. We have had a question, but I'm going to jump into it anyway. So what Newsflow can shareholders expect over the coming period?
So the coming period should be till the end of the year. We would be expecting to set up all of our manufacturing processes to be able to provide tablets for the trials coming up next year. Importantly, we'll be finalising dosing. We've got a good idea of what dose we want to take for the phase 3 trial in dogs with cancer, but we still want to optimise how we give that, how many times a day, whether we give loading doses at exactly what level, and then we'll be able to take that information into the human trials later next year.
Wonderful. Well, thank you for joining us, Richard, and a copy of today's recording will be available in the coming days on brokerbriefing.com. Thanks again.
Thank you, Jane.